So now we're going to move to the second talk by David Fawkner-Corbett. He's a clinical PhD student in the Simons Lab at Oxford University, and he's going to talk to us about the cataloging intestinal origins at sickle cell resolution with application to in utero disease. So David, unmute yourself and you can start pretty soon.
Perfect. Okay, thank you for the opportunity to present our work today. I'm David. I'm a clinical PhD student in the Simons Lab at the University of Oxford, sharing some of our work mapping human intestinal formation. The mature intestine is a highly complex organ with a huge diversity of cell types that is needed to form its various functions. At no time are the coordinated interactions of these cells more important than in development. After formation of the embryological gut tube, it's between 8 and 20 post-conceptual weeks that the intestine will go from a flat tube to more adult-like morphology. It is also at this time where a number of different processes that, if deviated from, will cause disease, such as the rotation of the gut. And many of the diseases happen around the first trimester, at which time it's almost impossible to collect tissue. Clinically, by 24 weeks, extremely premature infants can be born with relatively normal intestinal function, showing that the key lifelong circuits, such as the intestinal stem cell axis and the definition of small and large intestine, has already been defined. These circuits can become dysregulated in later life, such as in conditions as inflammatory bowel disease. In fact, work previously done in the lab has shown that single-cell RNA sequencing was a very powerful tool to chart the cellular diversity in IBD, showing specific subsets of fibroblasts and epithelial cells, including an unappreciated population of enterocytes. But this and other work in the field raises a number of important questions about intestinal development. Namely, when do specific cell types appear? How does a mature stem cell axis form? And what cell-specific in utero defects could contribute to disease? So to answer this, we collected fresh tissue from the Human Developmental Biology resource. We sampled from both the small and the large intestine, from samples from 8 to 22 post-conceptual weeks. We then used a dual hashing multiplexing approach to upscale previous efforts in order to generate a resource of 17 individual embryos from 40 tissue locations. This was also coupled with spatial transcriptomics in order to map the single cell signatures to tissue. In total, the cellular resource generated over 76,000 cells that divided into 9 compartments based on their transcriptional profile, representing the full thickness of the intestine. Even at this detail, it could be seen that clear differences were present in time, such as myofibroblasts appearing late in development, or in location, such as epithelial dividing into proximal and distal derivatives. Zooming in, we could then annotate all the cell states and subtypes, in total annotating 101 and their interrelationships. This is obviously a rich resource, but focusing on specific findings today, we first look at the intestinal stem cell niche. We're able to capture the appearance of mature intestinal stem cells in both the small intestine and the colon. We're able to see locational differences, namely within the proximal samples. There was a primitive population that preceded mature intestinal stem cell formation and transiently expressed a number of its markers that were lost over developmental time. We were also able to see the key stem cell marker LGR5 that was diffusely expressed early on in development and then became focal later in time, something that had previously been described in animal models. But even at the latest points in our resource, we saw that the intestinal stem cells greatly outnumbered that in adult data, both in single cell and in stainings, showing that the crypt, although looking more mature, is in fact still remodeling, which may be important for diseases of prematurity, such as necrotizing enterocolitis. Focusing on disease specifically, we leverage this data to link it with a human phenotype ontology and charted 749 disease-associated genes that are associated with a perinatal intestinal disease phenotype and mapped these to cell type specific effects. This led to a number of examples, such as RET, which is a well-known Hirschsprung's disease gene and could be seen around the myenteric plexuses in spatial transcriptomics and was expressed in neural cells. This differed to SOX10, another Hirschsprung disease that was expressed in glial, most highly, in fact, in a cell type that we annotated as lymphoid-associated glial due to its interactions with Peyer's patches, something which could have implications for Hirschsprung's disease. We could also see another example of an exceedingly rare DGAT1 deficiency that leads to severe malabsorption as seen in proximal enterocytes. But finally, we could use the time points and these effects to look at the time of specific gene expression. This example being HMGA2, which was only expressed in glial at 8 to 10 weeks, the time that the intestine undergoes rotation And indeed, this phenotype was associated with malrotation, showing how these could identify cells that cause rare diseases. So in conclusion, we carried out a single-cell and spatial transcriptomic study of human fetal intestinal development, capturing key developmental time points, which of course haven't had time to go into today, but I'm more than happy to discuss at the linked poster session. We linked this with the disease, and they're now publicly available. I'd just finally like to thank all the people that were involved in this huge undertaking, most notably Agni, who was key in all the steps of this work, and also the people that supervised, provided tissues, and funded the work. Thank you.